This is a production of Cornell University.
So good afternoon, everyone. My name is Mingyuan Zhu from Roeder Lab. It's a great pleasure to be here to present my work. But before I talk about my project, I want to do a quick test. So look at the two groups of the fruits and vegetables shown here. Which group do you prefer? So anybody prefer the lemons and the eggplants on the left? Please raise your hand. Oh, okay. So anyone like the apples with variable sizes? Oh, okay. So it seems people prefer more uniform agricultural products. So the shape and sizes uniformity indeed matters. So here is a key question in developmental biology and also the title of my today's talk. How do organs form with consistent sizes and shapes? So our lab uses Arabidopsis sepal as the model system, and the sepal is a green leaf-like floral organ. The reason why we choose sepals includes three major parts. Firstly, sepal development is relatively insensitive to the environmental changes. Also, we have plenty of samples for assessing individual plants. In addition, sepals are very easily accessible for experiments. So to study the mechanism about controlling the organ size uniformity, we firstly use mutagenesis to disrupt it. So we identified one interesting mutant, BOS2, short for variable organ size and shape 2. Here shows the phenotypes of the BOS2 flowers. So you can see some sepals are smaller than another ones. I also did quantitative analysis to further confirm the size variability. So on the right, it shows the sepal area distribution of the BOS2 is wider compared to that of wild type, indicating higher variance. So why the sepal is more variable in the BOS2 mutant, to answer that question, I firstly try to obtain some insights from the gene function. So I did MapBest cloning and identified a G2A point mutation happening in a gene which encodes MIP domain protein. And I tried to express the fluorescent proteins under the promoter of the gene and found an expression in the early flower, indicating the role of the BOS2 in the early flower development. So then with the help of the SEM, we try to find the stages when we can see the sepal variability phenotypes. So for the wild type, you can see the 4 sepal primordia evenly distributed around the boundary of the floral meristem. However, for the BOS2 flower, you can only see 3 sepal primordia. And as indicated with the red arrow here, 1 sepal is missing at this stage. And the sepal size variability phenotypes stay visible through late stages. So the sepal size variability phenotypes arise very early, and it indicates that the sepal initiation might be disrupted. To confirm that, I did a live imaging to track the organ initiation by imaging the same flowers every 6 hours. So here shows the representative time points from the live imaging. So for wild type, at the beginning, we can see the initiation of the first sepal, and within 12 hours, we can see the initiation of all 4 sepals. And after 42 hours, 4 sepals cover the whole bud. However, for the BOS2, again, from the beginning, we can see the initiation of the first sepal, but after 12 hours, still only 1 sepal visible. And after 42 hours, you can start to see the initiation of sepals here. So the initiation of BOS2 sepals is indeed delayed, and why is that? So it is proposed that plant hormone auxin and cytokinin regulates the organ initiation. So here I firstly use DR5 to check the auxin responses. So for the wild type flowers, we can clearly see 4 clusters of the DR5 signals, nicely correlated with sepal initiating positions. I then use the TCS to check the cytokinin responses. And again, we can see the 4 clusters of TCS correlated with the sepal initiating positions. So the auxin and the cytokinin balancing exists at the sepal initiating positions. How about for the BOS2? So the DR5 patterns become more diffuse in the BOS2 flower, and more importantly, the DR5 signal is much weaker. And the TCS signals almost form a ring around the boundary and seem strongly enhanced. So in the BOS2 mutant, the auxin cytokinin balancing is disrupted in the sepal initiating positions, which might account for the delayed sepal initiation. So here is a take-home message. We identified a MIP domain protein, BOS2, which regulates the auxin cytokinin balancing during the early flower development. The regulation through the BOS2 ensures the robust organ initiation pattern, which leads to the final uniform organ size. You can find more details in my EXIST seminar on March 22nd, and all of you are invited. And thank you very much.
So we have time for a couple questions. Can
That's
you repeat the question?
okay. So the question is only sepal affecting the mutant, or it also affects other organs such as leaves? So the answer is actually the leaf is much less affected than the sepal. But we also checked like the other floral organs like petal. So petal actually is also affected, much stronger than the leaves, but weaker than the sepals. But it seems like it's mainly, the phenotype mainly focused on the floral organs. Yes.
Plants have an interest to have very uniform seed size,
Yes.
and so they have lots of control mechanisms to make sure that seeds stay. So if you mutagenize, is it harder to disrupt to create variability in seed size than other organs?
I don't know whether it's more difficult, but I can answer with my mutant. So in my mutant, actually, the cytokinin and auxin disruption actually will even affect the formation of the seeds. So some of the seeds will be, we will say it might be even kind of embryolethal. So if we dissect the calyx, we can see some seeds, actually we can see they can not form normally. And for some seeds, they are maybe smaller. You can see that it's not really normally formed. So I would say this gene would also affect the seed formation and maybe also cause some seed size variability. So, but I'm not sure how, kind of the mechanism, yes.
Okay, thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.